One of the fundamental characteristics of life is that all life is based on genetic material. We now understand that genetic material is basically the molecule called the DNA, which is the basic blueprint for all life forms on Earth, and much of it is common to all life forms on Earth. But at some point, this was up to contention. A lot of people used to think that proteins were more important than DNA as the molecule of life, and we could still argue that point. Because proteins are more varied. They have a lot of different kinds of monomers, and they can be put together into different kinds of structures, and therefore generate different kinds of functions. All that you are is basically made of proteins, and these proteins do a lot of different jobs for the cell. And that's why it's kind of like, are you made of proteins or are you made of DNA? Which one is more important? You have to ask yourself though, would there be proteins if there wasn't DNA? But would there be DNA if there wasn't proteins? Since we're going to learn about in this video how much of the DNA structure and replication process is dependent upon proteins. And so there seems to be a catch-22 situation. But one thing is for sure, DNA is very important for life because almost every single example of life on Earth is based on DNA. And on top of that, modern biological research focuses a lot on comparing DNA from different people, from different species, and even historically, to try to understand what makes life the way it is. But this wasn't so clear to the scientists on the early 20th century, because even after Mendel identified the idea of a genetic factor and the hunt began to find it, DNA was an early candidate, but so was proteins. And even after we actually researched the idea of chromosomal inheritance and we looked at the different kinds of things the chromosomes were made of, we realized that as much as 90% of the chromosomes were made of proteins because of that protein scaffolding made of histones. And now we also understand that a lot of the things that DNA does requires proteins. And so came in the question, is it proteins that make us who we are? Is it proteins that's just a genetic factor, or is it DNA? But we now understand that DNA is definitely the key to make proteins, and that it is the genetic factor that Mendel was referring to. The DNA is what stores the genes. And we're going to now review the evidence that supports this, and at the same time discuss advancements that made us understand the structure and function of the DNA molecule as the central part of all life on Earth. It all started with Frederick Griffith in the early 20th century, and he got a mouse and it's injected with both a bad and a good bacteria. Well, I wouldn't say good bacteria, but a bacteria that wouldn't kill it. So the S strain would kill the mouse, but the R strain wouldn't do anything. The smooth strain was virulent or deadly. But if he actually got the smooth strain and heat stained it or killed the bacteria, it wouldn't do anything to the mouse. And yet, if you would get the pieces of that killed bacteria and put it in the same place that the R strain was at, then all of a sudden the R strain became deadly. Almost as something about the remains of the S strain were incorporated into the R strain and made it deadly. And this phenotypic transformation experiment proved that there was something about the pieces of the bacteria which were made it change. So there was the genetic factor that Mendel was talking about, and that was transferable. Avery, McCarthy, and MacLeod repeated this experiment, but first they separated the parts of the cell, for example, the DNA that you see separated in this vial, and they identified there was DNA that needed to be added to actually cause the bacteria to transform. Identifying the DNA is the key. The problem is that chromosomes were made mostly of proteins, so the answer was not clear yet. Not until Alfred Hershey and Martha Chase worked together working with bacteriophages, which are a special type of virus which attacks bacteria. And they tagged the molecules with dyes. And they tagged the proteins with a red dye and the DNA with a different color dye. And they noticed that only the DNA was necessary to go inside the cell to actually cause the virus to change the bacteria. If the protein never quite went inside the cell, proving again beyond definitive doubt that DNA was the genetic material, not protein. Then other scientists started studying the chemical composition and the structure of the DNA, starting with Aaron Shargraf, which actually studied the base pairing rules and the chemical composition of DNA. He realized DNA was made of phosphate, nitrogenous bases, and a 5-carbon sugar. And he realized there were 4 different kinds of base in DNA: adenine, thymine, guanine, and cytosine. And he noticed that adenine was always bonded to the thymine with 2 hydrogen bonds, and the guanine was always bounded to cytosine with 3 hydrogen bonds. And because of that specific pattern, we eventually would figure out that the length of the DNA molecule, or the width of the actual staircase of the double helix molecule, is constant because it's always a double ring bonded to a single ring. If it was anything else, the DNA would be too wide or too narrow. the fact that it's always 3 bases in the middle will actually make DNA consistent with the X-ray data which was collected by Maurice Wilkins and Rosalind Franklin. Maurice Wilkins was the scientist that invented a machine, the X-ray diffraction machine, that was used by several scientists to take pictures of molecules. And molecules that were helical in shape made the same kind of picture that DNA made. And so we knew that DNA must have been helical. And that's how we get to Rosalind Franklin and discovery that DNA was a helical molecule, and that made sense since you have always a base pair that's a double ring and a single ring, creating that pattern that we saw before. By the way, the guanine and adenine, which are double rings, are called purines, and thymine and cytosine, which are single rings, are called cytosines. And in the video where I go into this in detail, I actually teach you guys a mnemonic to help remember this. Then came other science, which tried to explain everything the DNA did with different kinds of models. He suggested a triple helix model. He suggested parallel strands folded out models and a single helix model. But none of those models actually explained everything the DNA did. And the race was on to kind of win the Nobel Prize, because it was announced that that would be done as soon as the first person came up with a model that fully explained everything the DNA does. The winners were James Watson and Francis Crick, which came up with a double helix model of the DNA molecule. You see them working at it in the bottom there. And the double helix model explained everything that DNA does. And we'll review that momentarily. But before that, there are actually 2 kinds of nucleic acids, DNA and RNA. The major difference between RNA and DNA is that DNA is double-stranded, and it contains all the genes in your body, while RNA contains only a copy of one gene, so it's much shorter, and it's also single-stranded. Also, the strand in RNA does not contain thymine. It's replaced with uracil in RNA for stability purposes. It also has purines and pyrimidines and the same kinds of relationships that you would find in a DNA molecule. Another big difference is that the sugar is going to be different, because the RNA nucleotide contains ribose, not deoxyribose, which is missing oxygen on the second carbon, as you see in the picture here. And they also have different functions. DNA stores all genetic information, and RNA is mostly involved with transferring that information into actual protein. Also notice there's a very close relationship between the nucleotides found in DNA and the molecule of ATP, because nucleotides are basically made of phosphate, a 5-carbon sugar, and a nitrogenous base. And that's exactly what ATP is made of, except with the 2 extra phosphate groups. And we actually call that the nucleoside triphosphates. And that's what actually is going to be used as the building block to the DNA and RNA molecules. And so there's a very close relationship between the energy molecules of the cell and the nucleotides which make up the DNA code. And that's actually going to be important when we talk about how DNA is strung together and how life actually recycled this molecule for that purpose. Let's review the basic structure of the DNA molecule. The shape of the DNA molecule is a double helix. It's got a backbone made of phosphate and sugar strung together by phosphodiester bonds, and the rings of the latter are made of nitrogenous bases, which are always paired according to Chargaff's base pairing rules. Guanine with cytosine and thymine with adenine, and always 2 bonds between A and T and 3 bonds between G and C. And to make those bonds happen, DNA must be in an anti-parallel direction, which means one strand will go from 5 to 3 and the other strand will go from 3 to 5. And you get those numbers by counting the orientation of the sugar in a molecule. Notice how one sugar points up while the other sugar points down. That's the only way that the base pairing rules will actually happen, if this anti-parallel strand actually happens. And in DNA, most of the bases are A and T, because A and T are part of the promoter boxes and centromere regions and telomere regions, which make structural things and regulation factors for the DNA. And that's why in human DNA, A and T are more common than G and C. 60% of DNA is shared between A and T, 30% each, and G and C share 40%, 20% each of the nitrogenous bases in DNA. That's a discovery that was, by the way, made by Ibn Chargaff as well. The base pairing rules also explain why the distance between the helixes are always constant at 2 nanometers. And the vertical attraction that exists between the base pairs are also going to explain why the DNA molecule twists. It twists for stability because there's vertical attractions between the molecules. And therefore, it will complete one full twist every 10 base pairs, or 3.4 nanometers, since there's a vertical distance of 0.34 nanometers between each base pair, or between each nucleotide. And that's constant because of the phosphodiester bond that connects that backbone. By the way, the hydrogen bonds is what connects each strand of the double-stranded DNA molecule. DNA normally twists to the right side. If it twists to the left side, that's odd. It's called zDNA, and it will cause mutations to the DNA copy process if that's actually happening. You should be able to draw this or explain the shapes of the DNA, and you should be able to review what gives us the evidence for the shape of the DNA molecule. We know that it's a twisted double helix because of the base pairing rules and the diffraction X-ray pictures of the DNA. We know the width is always the same because of the base pairing rules and the hydrogen bonds that make them. Always 3 rings in the middle. That's why it's always the same thing between the distance between the strands. We know the base-to-base length is always 0.34 nanometers because of the phosphodiester bonds that make up the backbone. We know that nucleoside percentages are going to be the way they are because of the phosphodiester bonds and because of the composition of the centromeres, telomeres, and promoter sequences of the DNA molecule. We also know that each DNA will be twisting because of vertical attractions between the molecules, which gives them more stability to twist, and that it's always going to be 10 base pairs per turn, 1 large groove, 1 small groove, completing a turn every 3.4 nanometers. And we know that the direction of the twist is going to be normal DNA or right side of DNA. Left side of DNA is going to be called zDNA or southpaw DNA, and it usually will cause mutations. Now, that double helix will actually call itself up and form chromosomes or chromatin, which is what you actually find inside the cell. And this calling will happen around histones, which make a protein scaffold, which actually makes 90% of the composition of chromosomes. These histones gather together to make nucleosomes, which then coil into coils, which then coil into supercoils, and then into the actual chromatin fibers that make the chromatin or the chromosome. If the chromatin is opened up and being read during protein synthesis processes, we call that heterochromatin. If it's a little more coiled up or inactivated, it's called euchromatin. And euchromatin coils even further to form chromosomes during the cell division process, and they're visible by the end of telophase. There you see, by the way, the difference between chromatin and chromosomes in this particular drawing. Now, chromosomes have a long arm and a short arm separated by a centromere. The short arm is called P or petite arm, and the long arm is also called Q arm. Now, normally a chromosome is only a single linear DNA molecule that's coiled up in that system that I just described. But during the S phase, the DNA will double up so that by the time you get to telophase, you don't see the singular chromosome DNA that you should see if the chromosome were to coil during the G1 phase when it's normal amount. You will actually see the X-shaped chromosome bound together by the centromere if you were to do that. And each one of those exact copies is called a sister chromatid, and as we reviewed in the cell division lectures, that's going to actually separate during the cell division process to give each daughter cell exactly the same amount of DNA. Also remember, of course, that the human karyotype includes 23 different kinds of chromosome or chromosome pairs. 1 copy we see from mom, 1 copy we see from dad. And we have 22 of those which are autosomes, which determine our normally body functions, and 1 which is the sex chromosomes, which help determine the sex characteristics as well as other functions as well. If you're XX, as you see on the bottom right, you're going to be a female. If you're an XY, as you see in the bottom left, you're going to be a male. Karyotypes like this have been created by several different methods, including the fish method, which involves centrifuging and tagging the DNA to separate the chromosomes into different sizes. And you actually make a karyotype by lining up chromosomes of different sizes into family groups like you see in these pictures. That's a review of the history of the discovery of the structure and function of the DNA molecule. And on the next video lecture series, we'll talk about how the DNA is actually copied.